Hello, my name is Professor Matthew Schmidt, and welcome to Genetics. Today we're going to take a look at some of the earliest history of genetics, particularly the idea of how information, which is really what genetics is, biological information. We want to know how information gets passed down from parents to offspring. Throughout the ages, just by looking around and not being very scientific, it's clear that parents give information somehow to their children if you're talking about humans, and the same goes for most animals and even other types of organisms. We're calling today's lecture Mendelian Genetics, as it was named after the person who's often referred to as the father of genetics, Gregor Mendel. He's the first that gets credit for actually doing experiments, keeping detailed records, and really proposing a theory about how this happens. So technically, we're going to look at monohybrid crosses, which he did, and he came up with a law that he called the law of segregation. Sometimes we call it Mendel's first law today. So let's start by seeing some general historical facts and ideas about genetics before Mendel, because for most of human history, let's face it, people knew very, very little about how information was transmitted from parents to offspring. So as I mentioned, Gregor Mendel is often referred to as the father of genetics. Before Mendel, and he did his work in the mid to late 1800s, almost nothing was known about genetics. There were observations made even by non-scientists, just common sense observations, that led people to believe in a theory that they called, and we still call today, a blending inheritance type of an idea. You can think of blending inheritance in the following way. It's as if whatever information is being donated by a father, for example, people didn't know what that information was, obviously, but some information was coming from the dad, some information was coming from the mom. And you could think of it as almost literally getting mixed up in a blender, just homogenized into some kind of hybrid type information, and that's what ended up in the offspring. The reason it was thought that this was the case is because you can make observations, well, you could make them now, and you could have made the same ones then. For example, if a very tall individual was to have a progeny with a short person, then the offspring very often had a height that was somewhat intermediate in between the tall and the short, sort of blending them together. If someone with very dark skin had an offspring, a child with someone with very light skin, by the same idea, very, very often the child's skin coloration was somewhere intermediate between the two parents. And you can think of a lot of examples like this. So it wasn't really crazy to think about blending inheritance. In fact, a lot of real-world examples seem to give credence to that idea. Well, Mendel was the first to really do some very strict experimentation based on sound principles to keep very good records, and he proposed theories of inheritance that actually made sense and, very importantly, were testable, and we can almost do the exact same type of crosses that he did today. So I guess it's logical to ask and answer, what exactly did he do? Well, firstly, Mendel chose to use the pea plant, the common garden pea of the genus Pisum. And why did he use that? I mean, he wanted to get at some general principles of genetics. At the time, of course, he didn't know if the genetics of the pea plant was going to be different than that of the fruit fly or whoever else. But basically, this may seem silly, but it's true. They were available to him. He had some pea plants. He was, as the story goes, an Austrian monk he was a teacher, but he also took care of the garden, and he was very into his plants. So he had peas, so why not use them? Very importantly, peas, as well as most other plants, can either self-pollinate or they can cross-pollinate. What this means is that unlike humans and many animals, most plants have both the male and the female sexual organs together in the same flower. So you can take roughly the equivalent of sperm, which is pollen from a plant, and you can put it right from the same plant, pollen, right onto the receptor for the egg. And basically it would be the equivalent of, this sounds very strange, but of a person literally having sex with themselves from a genetic perspective. Obviously, we can't do that, but pea plants can, and you'll see that this is very important to the experimental design that Mendel came up with. By the same token, though, you can cross-pollinate them, which would be a sort of more traditional sexual exchange of information where the pollen from one plant is used to fertilize the eggs of another plant. So it's very important that that can be done, the self and the cross-pollination. Also, they produce many offspring. So when you do a statistical analysis, you may know that the more data you have, the better off the results are. Just to use humans as an example, humans can have a lot of kids, but a lot might be 7 or 8, right? We're not talking hundreds. So it's better to have more data when you're trying to calculate results and see what they mean. Peas also have a short so-called generation time, and what that means is the amount of time from fertilization to the sexual maturity of an adult plant. So in peas, that's about 2 weeks. Again, to use humans as an example, our generation time is about 15 years. So we would have a long, long wait if we wanted to analyze all of the offspring of a particular cross. So for all of these reasons, peas were attractive in terms of being good for experimentation. And the last one may sound a little bit silly, but it's very, very true. Unlike animals, when you want to try and get them to breed and count them, they don't run away and say, I don't really want anything to do with this. So if anyone's ever worked with fruit flies, which are a model organism for animals, you have to anesthetize them when you want to count them. And unless you do it very well, often they'll wake up and sort of fly away when you're in the middle of counting, and that can really compromise the experiments. Plus, they'll breed, but you can't actually force them to. In the case of peas, you just take a little paintbrush, take some of the pollen and wipe it onto the female part of the flower, and then you've done a fertilization event. So these, among probably even other reasons, are why the pea plant is a very, very good experimental organism. Now, Mendel studied 7 traits altogether, and he used these traits very specifically because always these traits could be found in 2 forms, and only 2 forms, in the pea plant. So just as examples, pea plants have flowers that are either purple or they're white. There's no red, yellow, green, nothing in between, purple or white. Mendel used the term, he called these antagonistic traits or antagonistic pairs. That terminology really is not used anymore, but if you happen to see it, means it's really either or. You're purple, you're white, and those are the 2 variants. Unlike in people, pea plants are either tall or short. I don't know the exact height, but in other words, there's not this range of heights. It's tall or short, one or the other. Green seeds versus yellow seeds, and so on and so forth. So the bottom line is there's always 2 variants of the trait, and it's one or the other that the trait comes in. So his experimental rationale is extremely important. Guys, this is the bedrock of everything. If you don't understand this part, it's hard to go on to the further parts, which is why we need to spend a little bit of time on this, or perhaps a lot of time on it. So we're going to use flower color as our example, not for any particular reason except that I like flower color and it's something very definite that you can see and keep track of. So the flowers of the pea plant, Pisum, could be either, as I said, purple or white. That's really pretty much what the pea plant flowers look like there. And remember, there's only 2, these are the only 2 possibilities with respect to pea plant flower color. Now, one very important thing to understand is that the whole basis of starting out the experimental rationale here is that there are pure breeding lines that either exist or can be created. Sometimes these are called true breeding. If you hear that term, it means the exact same thing. Now, if you have a pure breeding line of purple flowered pea plants and a pure breeding line of white flowered pea plants, what this means is, let's take the purple line. If you self them, and the term self means to self-pollinate or self-cross. In other words, if pollen from that purple plant is used to fertilize itself, the offspring will always be purple. And the same goes if that pollen is used to fertilize any other member of that true breeding line. So what Mendel did actually was observe these lines for literally years and say, you know, every time we make a cross with these plants that they always have purple flowers. So this is true breeding. They breed true, they'll always make purple, and the same goes for the white. So you sort of keep these apart from one another. We know we're always going to see purple on the one hand and white on the other hand. As we go through this, we're going to introduce some terminology. So the appearance of an organism with respect to a given trait is known as its phenotype. So if we said the flowers on this pea plant look purple, we're describing its phenotype. Looks white, we're also describing its phenotype. Now, we also want to see some of the conventional terminology with respect to the way crosses are drawn. And you can see here it starts out pretty simple, but what we're going to do in understanding what a pure breeding line is basically just to say that if we have a purple parent, and usually the X, letter X, or sort of a times sign is used to say we've crossed a purple flowered plant with another purple flowered plant. If it's a pure breeding line, the result is always going to be that the offspring are going to be purple. Okay? And you've probably guessed that if we do it with a white member of a pure breeding line times another white one, all of the offspring are going to be white. Okay, so keeping those ideas in mind, let's go on to the next part of the experimental rationale. And this is really where a fundamental question comes into play. So Mendel reasoned, he said, okay, I understand how true breeding plants sort of operate and how to predict what the results will be from them. But what if I took a member of a pure breeding line that was purple and crossed it with the pure breeding line that was white? If blending inheritance, as we discussed, was true, you might expect, and I suspect Mendel did expect this, that the offspring's phenotype, and by the way, when you start keeping track, that's known as the parental or the P generation. Okay, so the cross that we're doing here is a parental purple times a parental white. And the question is, what would be the prediction of the offspring? Now, if blending inheritance is true, the color would be somewhere in between the two parents, right? Now, I realized after I did this, but I left it anyway, probably somewhere in the middle between purple and white isn't really pink. It would probably be something more like a light lavender or something like that. But the idea of pink was just something in the middle between purple and white. And also I should add that these flowers that I grabbed to put on this slide are neither the right color nor are they pea plants, but you get the idea, right? So in other words, the prediction was probably that the offspring would have a color somewhere in between that of the two parents. That's the traditional idea, makes sense, but that is not what Mendel saw ever. And this is where the beauty and the elegance of his experiments start to come into play. So what were the results? Well, it turns out all of the offspring had purple flowers. To continue with our terminology, these offspring are known as the F1. That stands for the first filial generation. And just for the record, you can go down, as long as you want to keep track of these, to the F2, the F3, the F4. You can keep track of the generations. But right now we're interested mostly in the F1. Now look, all of the F1, I mean all of them, were purple. And it's the exact same purple color that the parental purple flowered plant had. So Mendel probably thought, I'm guessing, that's strange. It seems like I had a purple and a white, and now the whiteness seems to have gone away. It's disappeared. Now all I got is purple flowers. So it's not immediately clear what this means. This is data. It's an observation, right? So as the slide says, it's time to investigate further about what's really going on here. Now, here's really the experiment that brought home some of what seemed quirky at the time, but that Mendel used to formulate his law of segregation. If the purple F1, in other words, those offspring that were all purple that we were just discussing, are either selfed, in other words, self-crossed, it's the same thing as being crossed with another one of the F1, who are brothers and sisters. That's not something that we do generally with humans, but with peas it's okay. So what will happen? So again, if you look at the cross that we're going to do, we're crossing F1 purple times F1 purple. And what is the prediction? I mean, it's hard to say, but before, when we crossed a purple with a purple, we got purple, right? So it's not crazy to think that that's might be what happens again, because the white seems to have just disappeared for unknown reasons. Now, are they all purple if you do this? And boldly you can see that the answer is no. Here's the very specific data that Mendel observed in the F2 generation, which are really, if you want, the grandchildren, the second generation of offspring from where we started from. He observed that the F2 generation produced offspring that included both purple and white flowered plants. Those purple and white colors are identical to the ones of the original parent. In other words, there's never been any change in the actual purpleness or whiteness. They're the same. But he also noticed very definitely that there were more purples and not just some random amount. What's become famous is he detected a phenotypic ratio of 3:1. In other words, for every one white flower, there are 3 purples. There are a few ways you can say this. You could say that 3/4 of all of the offspring were purple and 1/4 were white. Most traditionally, we just say we have a 3:1 ratio of purple to white flowers. Now, this is a very definite thing, 3:1, and it's not immediately obvious why this is happening. So Mendel had to go think about this for a while, and I think we should look at a sort of graphical depiction of this so-called monohybrid cross. So the crosses he did at the beginning were monohybrid, and that simply means that we're keeping track of one trait, in this case flower color, as we're going along doing the cross. Remember, these pea plants have all sorts of other traits. They're tall, but we're not worried about that right now. We're only keeping track of the flower color, thus a monohybrid. Someday in the future we'll see dihybrids and maybe even trihybrids. But basically here's what we did, right? Just to sort of sum it all up and make sure we understand. In the pea generation, we crossed a pure breeding purple times a pure breeding white, and in defiance of perhaps what we might have expected, we saw that all of the F1 offspring were in fact purple, so all purple. Then what did we do? We basically crossed one of these with itself or its brothers or sisters. These are all genetically the same. Well, he didn't know that, but they're certainly all phenotypically the same. Now let's look at that cross. So we took a purple from the F1, another purple from the F1, and instead of them all being purple, we found that they were purple and white in a very, very definite and distinct ratio, which was a 3:1 ratio. Okay, so that's sort of actually sort of pretty graphical depiction of what Mendel did. Now, before we get into analyzing what it means, just for the record, before Mendel figured out what it meant, he did the same exact experiment with 7 different traits and always got the same results. So in other words, what I mean by the same results is if you take pure breeding lines, let's say tall and short, cross them together, the F1 always resembles one of the parents in the exact same version of the trait. In other words, if you happen to do tall and cross them with short, all the F1 are tall. All right. And then if you self the F1, you always get a 3:1 ratio, in this case of tall to short. So the point of this is to show that the flower color experiments are not just some isolated weird thing that occurred, that this is a general principle that occurs no matter what trait we're looking at. Right? So I doubt that you would have to memorize every single one of these crosses that he did. It's possible. But for the record, these are what they are. So seed color could be yellow or green, and if you cross pure breeding lines, you see that all the F1 were yellow. This pattern, hopefully, it'll just become clear, this type of experimental pattern, just the same over and over, but that's good. It means this is a general principle. The seed shape could be round or wrinkled, and all the F1 were round if you did it, the experiment the way we're talking about. We already did flower color, but you see that all the F1 were purple there. And very quickly, just pod color could be green or yellow. In this case, all the F1 were green. The pod shape, you could sort of have what they called round or pinched. All the F1 were round. Stem length, we discussed tall versus short. All the F1 were tall. And also the flower position, they could be axial or terminal on the sides or at the top. All the F1 were along the sides. Okay, so I think the idea here more is not the traits themselves, but it's the fact that it always works the same way. All right. Now, let's interpret these results. And this is very, very important. This is what Mendel, after really looking at the data, said, okay, this is what's going on. So we call, perhaps obviously, we say this is Mendel's first law. He couldn't have named it his first law because he really didn't know he was going to come up with second law. He called it the law of segregation, but it states all of the following, and I can't overestimate, or I should say overemphasize the importance of these things. So what he said is, look, for each trait, say flower color, there are discrete pieces or units of information which he called factors. Now we're not going to use that term again because Mendel's factors are what we now call genes, and almost everybody knows that term. So basically he's saying that, look, a gene is a unit of information about flower color. Discrete, that's what it's about. And furthermore, that every adult plant possesses 2 copies of a gene for each trait. So with respect to flower color, whatever color you are, you're going to have 2 units of information, 2 genes about that trait. Now here's a very important part, and it has to do with our whole understanding of a sexual life cycle. During the formation of gametes, and gametes means the same thing in people, plants, or anything. Gametes are sex cells. They're usually sperm and eggs. Technically, plants do make sperm, but they're not the same, obviously, as animal sperm, so they're pretty much like pollen grains. We'll get into more detail about that later, perhaps. But the idea is, when you're making gametes, whether you're a person or in this case it was a pea plant, he said that during the making of these things, that the 2 copies that you have segregate or separate, and one goes into one gamete, one goes into the other gamete. That means that each gamete only has the 1 copy, all right? Now, since they have the 1 copy, what puts them back together again is the fertilization event, okay? So if you have 2 gametes each that have 1 copy of a gene and they go together, then the new fertilized, technically it's a zygote, is going to be back to having 2 copies of the gene for that trait. And what that thing really is, it's an embryo that's in a seed that will then grow into an adult. So we've taken the 2 copies, separated them to make gametes, and then put them back together to make a new plant. And if you understand this, this allows us to interpret the results of our experiments. Before we can do that, though, he had to postulate some more ideas and some more terminology that goes with them. So one very important thing, and perhaps obvious thing, is that genes come in 2 different forms called alleles. So this term alleles is very important. What it basically means is you can think of it as this. There's a gene for flower color. In other words, a piece of information about flower color. But there are 2 versions or 2 flavors, if you will, of this. And there are 2 different alleles because one of them is saying, okay, with respect to flower color, be purple. That's the purple allele. One is saying with respect to flower color, be white. That's the white allele. So if you ever get confused about allele, think version. The white allele and the purple allele are both different versions of the flower color gene, the unit of information about flower color, okay? And you also had to postulate that one allele is always dominant to the other, and the one that's not dominant is known as recessive. There's a couple ways of thinking about dominant alleles, but the best definition, I think, is that an allele is dominant if the version of the trait it controls, in other words, purple flowers, shows up when 1 copy of that allele is present. That means there might be 1, there might be 2, it doesn't matter. As long as there's 1 copy there, the trait is going to show up. So we're going to see that if a pea plant has at least one purple allele, the flowers are going to be purple. With respect to a recessive trait, both, remember there's two copies, both copies have to be that recessive allele if you're going to see the trait that controls. In the case of flower color, that would be white flowers. So an important term that goes along with phenotype is the actual combination of alleles present in an organism constitute its so-called genotype. All right, so we're going to see as we now look at, there is a couple more terms that we need to be aware of, but this is all going to come together as we revisit the experiments and see what was really going on. So also, since we now know that each organism has two genes for two alleles for each trait, if you have two of the same, we're going to say that you're a homozygous organism or simply a homozygote. You may know that the term homo means the same, and that's exactly what's being said here. Just be a little careful, because if you have two purples, you're homozygous. If you have two whites, that plant is also homozygous. Since purple is dominant to white, a purple homozygote would be referred to as homozygous dominant, whereas the white would be referred to as homozygous recessive. If you have two different alleles, and in this case that would mean one purple and one white, that's really the only way you could have two different ones, you're then called a heterozygote. Hetero means sort of other or different, and that's what this means here. So you're heterozygous or a heterozygote. Now Mendel didn't really use this term, but this is really an opportune time to sort of throw it in there. A gene's locus is the physical location of that gene's information on a chromosome. Now it's important to realize that Mendel never heard of a chromosome, I don't think, in his life. If he did not make any connection between his work and what a chromosome was. We now know, and we knew for about 100 years or so, that the genes Mendel discovered are in fact located on chromosomes. So furthermore, they have a very definite position, literal physical position on the chromosome, and that's called the gene's locus. So I'm just making up the specific position right now. We could look it up, but let's just say that the gene for flower color is located on chromosome number 7, right at the very end of the right arm or something like that. For those of you who know, that terminology is not perfect, but my point just is, what you would say is, look, regardless of what allele is there, information about flower color always exists in peas on chromosome 7 toward the top. The locus, think of the location. That's where it is. So you could make a statement as follows, and if you need to rewind this and listen to it a couple of times, it's important to make sure you understand how these terms all fit together. So I could say that at the flower color locus in this particular pea plant, there exist two genes, and one of them is the purple allele and one of them is the white allele. Therefore, this organism is a homozygote. Sorry, it's a heterozygote. I almost confused myself. Because there's one purple and there's one white, and this is a heterozygote, and because purple is dominant to white, the phenotypic appearance is going to be that this is purple. Genotypically, as we just said, it's going to be a heterozygote. So just think locus, at this location, that's where that information always is. And what is the information? Well, it could be purple. It could say purple, which would make it the purple allele. It could say white, which would make it the white allele. The purple or white allele of the flower color gene, which always exists at that locus. I'm sorry if that was a bit much for that, but these terms are often confused. Okay, so now, with all this under our belt, we and Mendel can understand the results of our experiments, or more technically, his experiments. So I'm going to show you the same sort of summary of the crosses that we did, but now with the genotypic information. So remember what we did. Look, we had a pure breeding line of both purple and white flowered plants. Now we know why they were pure breeding, because look, guys, a pure breeding purple plant is in fact pure breeding simply because it is homozygous dominant. That is, what the two copies are in that plant are both purple. Now, since purple is dominant to white, the convention is we draw the dominant gene as a capital letter, in this case capital P. So purple flowered pure breeding plants are capital P, capital P. And it doesn't take a lot of analysis to think about it, that if these keep being put together and put together, all there are capital P or purple alleles. You're never going to get anything else, no matter what. And that's why they were pure breeding. And the same goes for the white. Those are little p, little p. Now, little p is the white allele because it was named after purple, but purple is dominant to white. So again, if you keep crossing these pure breeding white plants together, all there are is little p. You're always going to get white. Now, we're going to do a thing called a Punnett square in just a second, but I want you to see here, it doesn't really take a lot of thought to realize that, look, when gametes are being made by this plant, all it can donate is the purple allele, the big P, right? All this one can donate is the small p or the white allele. It's pretty clear that all of their offspring are going to have one big P and one little p. So their genotype is going to be that they're heterozygous, right? But phenotypically, since purple is dominant to white, big P is dominant to little p, we already know all of these are going to phenotypically appear purple, just like that original parent plant. There's no dilution, okay? So what you would want to take away from this is that while the phenotype of the F1, the appearance, is identical to one of the parents, the genotype is not. All of the F1 are heterozygotes. They're big P, little p. They look purple because of the dominance. And this is going to explain everything. When we now, remember the next cross we did was to take one of these and cross it with either itself or one of its brothers and sisters. So now when we go ahead and look at that, we'll do the Punnett square in just one second. When we go ahead and look at that, we have to realize that our F1 purples are heterozygous to start with. So even though they look just like the original parent, they're phenotypically purple, their genotypes are not the same. All right? And we're going to return to this in just one second. Let's go back and just understand how to do a Punnett square. We're going back to the original cross here of the pure breeding purple parent and the pure breeding white parent. So you guys know that the genotype of those was big P, remember that's why it's pure breeding in the first place, purple, times little p, right? It's important to know how to do a Punnett square. So this one seems a little bit redundant, but what you're saying is, based on the idea of separation, one big P goes into one gamete, the other big P goes into the other. And we want to put the gamete types on the tops and the sides of the Punnett square. So for this one, we arbitrarily put them on top. It sounds a little silly because what we're saying is, being big P, you can make either big P gametes or big P gametes. I mean, that means you can only make big P, but it's good to keep a common form, as you're going to see when you're doing Punnett squares. By that same logic, this plant can only make a little p or a little p gamete, right? Which means, again, they're all little p. But the way a Punnett square works is, you put these things together, you have to think of it this way. So if the big P from one parent is meeting up with the little p from another parent, all of those offspring are going to have one big P and one little p, and they're going to be heterozygous. And without belaboring the point, you can probably see that that's going to happen in every one of the boxes of this Punnett square, right? If this big P meets that little p, if this big P meets that little p, and if this big P meets that little p. They're all different events, but the outcome is the same, which means that all of the F1 are purple, but they're heterozygous purple. So when we go to do our cross here and fill it in, we have our two purple plants. They're the same ones from that Punnett square that we just made. We're going to do a Punnett square about this, but what I want you to think about is, this parent can make gametes that are either big P or little p, and the same goes for this parent, right? So it's going to make our Punnett square a little bit more complicated. Now you knew from before that we saw a 3-to-1 ratio of purple to white in the F2. What I want to bring your attention to here is that even though all three of these look exactly the same, that is, they're phenotypically purple, genotypically they're not the same. One of them is homozygous dominant, big P, and two of them are heterozygotes. Because of the dominance of the purple allele, remember our definition, as long as one big P is there, it's going to be purple. So yeah, one big P is there in each of these. It doesn't really matter what else is there. That's really what dominance means. However, and probably this makes sense, the white flowered plants, the only way they could be white flowered is to be little p, or homozygous recessive, right? So let's do a Punnett square that would allow us to predict why this 3-to-1 ratio comes about. So we're going to do it the exact same way, just realizing that each of these parents can make either a big P in one of their gametes or a little p. There's 50-50 chance of either. And the same exact thing goes for this parent. But now when we fill in the boxes, look what happens. So here a big P is meeting up with a big P. That will create an organism that is big P, or homozygous dominant. Because there's at least one big P there, it looks purple, which is why we colored it in purple. Here a little p is meeting up with a big P, creating a heterozygote that also looks purple. Here a big P is meeting up with a little p, creating a homozygote as well. These two, this one and this one, are genetically identical, but they do represent two different events. In one case, the little p came from the sperm, and in one case, the little p came from the egg. So we have to count them twice because probabilistically they do represent two different events. And I think by now you're getting that if two little p's come together, that's the way and the only way that we're going to get a white organism here. All right. So before you did it, you can use this information. Mendel got the data first and then figured this out. But now that you know it, you can predict by doing a Punnett square what proportion of offspring will be different genotypes and different phenotypes. And a lot of problem solving will go to those type of questions. For now, though, we want to do is make sure we understand this concept of the monohybrid cross, the Punnett square, and all this business. So what we did is made a diagram that sort of summarizes the entire cross from start to finish. And we decided to use seed color in this case, not particularly because that's desirable or important, just to emphasize we've been using flower color all this time. You don't have to. You can use anything. Well, when I say anything, I mean any of the 7 traits and probably more that he studied that work this way. So in this case, let's take a look at what's going on here. We're starting with, you can see that one's yellow and one's green. Hopefully you now understand that because you can see one is big Y, that's a pure breeding yellow seeded plant, while the other one is a pure breeding green seeded plant. You could do a Punnett square, but clearly what this is saying in summary is the only gametes that yellow one can produce have a big Y or yellow allele in them, and the only gametes that the green one can produce have a little y or a green allele in them, right? So that means all the F1 offspring, as it says here, are going to be identical in their phenotype, which is yellow, and in their genotype, which is that they're heterozygous, big Y, little y. But since in this case, big Y yellow is dominant to little y green, they all look yellow, right? But now we're going to do the F2 Punnett square. So we've realizing here that this parent can make big Y or little y, right? The same goes for the other parent, showing one being the pollen and one being the eggs. Technically, it doesn't matter which one is on the top or on the side, but it's not a bad idea to decide how you want to do it and to do it the same way all the time. So when these meet, we've got big Y, right? A homozygous yellow seed. Here we've got big Y meeting up with little y, and here we've got a similar thing happening. We purposely wrote it this way here. Whether you say little y, or big Y, little y, Genetically inside of a cell, it's the same thing, but they're two different events, and you can see that here. Now, only when the two little y's meet up, you get a green homozygous recessive seed, and you can see from that box very clearly the 3-to-1 ratio, right? However many offspring there are here, 1/4 of them are going to be green, 3/4 of them are going to be yellow. So hopefully that really drives home the point of the way Mendel did these crosses. Now, the last thing we want to cover in this portion is the idea of a test cross, because Mendel used test crosses and they often come up in problems. So by definition, the test cross is a cross between a plant that is displaying the dominant characteristic, in this case yellow, and what you really want to do is to determine the genotype of that plant. Because let's face it, if I just said, gee, here's a yellow seed, what's its genotype? You know its phenotype, which is yellow, right? But the genotype is not known, so we're going to call this our mystery plant. It's yellow, but we don't know if it's homozygous or heterozygous. It turns out that the way you can find out is quite easy, and that is to cross it to a plant that's expressing the recessive characteristic. The whole key to this is, if I show you a green pea and you understand the dominance relationships, then you do know the genotype of that pea, right? It's little y, little y. So you have to do a little detective work, and you have to say, look, I'm going to cross the yellow with the green. Based on what the real true genotype is of our mystery plant, we're going to figure that out by looking at the offspring. So you have to get two scenarios going. What if, in fact, that mystery plant is homozygous? Well, if you cross it with the green, we're putting the Punnett square a little bit shorthand here. This can only make big Y, right? This can only make little y. So if this was the case, all of the offspring would appear yellow because they would be heterozygous, right? So if you did the cross and you saw that they were all yellow, you're going to go back and say, okay, well, look, this must have been big Y, because that's the way I saw the offspring. On the other hand, if that mystery yellow plant was heterozygous, half its gametes would contain big Y and half of them would contain little y, right? The green plant still can only make little y. And you can see here that what we get in this case is 50% yellow heterozygotes and 50% green. So here's the bottom line. If you do the cross of yellow times green, and say you observed about 50% of the offspring being green and 50% being yellow, you would go back and say, aha, that original parent was a heterozygote itself. Because if it was a homozygote, we would not have seen any green in the offspring. We would have seen only yellow. So that's what a test cross is. And you need to know, you need to memorize this. You can't memorize the concept, but my point is, if on an exam they say a test cross is performed, you're expected to know exactly what a test cross is and what it's designed to determine. And hopefully that's come across here. Well, thank you very much for joining me for this genetics session. I hope it was as exciting and fun for you as it was for me, and I'll see you next time.